Hello, everyone. My name is Cathy Roesch. I am Director of Science and Quality at Basque Engineering and Science. Thank you for joining us today in our webinar. Today, we're going to be discussing our technology, which is frozen sample aliquotting. Our main claim to fame is that we try to keep our samples, your frozen samples, frozen while working with them in order to aliquot out specific volumes to be used in downstream assays, because thawing samples is just not cool. So I'm going to talk today a little bit about frozen sample aliquotting. What is it? Coring frozen animal or human biofluids or tissues using nuclease-free probes to extract out specific volume aliquots without any thawing or contamination of samples, producing high-quality samples for downstream assays. What are the benefits of frozen sample aliquotting? The benefits are that it improves sample integrity and preservation. It eliminates sample contamination. It increases sample utilization and reaccess for later on. It allows scientists to work with their samples safely. It delivers sample consistency and homogeneity. It produces reliable results, data that is reproducible, and it's time efficient for scientists. So I'm going to go over each one of those individually. So how does frozen aliquotting improve sample quality? Well, it eliminates the freeze-thaw cycle that can degrade analytes, causing biased data in downstream analysis. Thus, it's literally stabilizing labile molecules because it keeps all the samples at -80. The second thing that we're going to talk about is it prevents sample contamination. Our patented technology uses nuclease-free probes to extract out aliquot, or cores, as we call them, from frozen biosample, free of contamination. Both the parent and aliquot samples are maintained at a temperature of -80 or colder throughout the entire process, ensuring well-preserved, high-quality samples for use in downstream assays. Number 3, it increases sample utilization and reaccessibility. One or multiple specific size cores are or can be extracted out, leaving behind an intact parent sample. The parent sample is then returned to the -80 freezer with reuse later on, free of contamination. Homogeneity. So let's talk about homogeneity. So homogeneity is really going to depend on how the sample was prepped before it was frozen. As we all know, different types of molecules, analytes, due to their size and density, tend to settle out in different places within the tube during the freezing process. So the coring process is always done from the top of a sample to the bottom of the sample, center stage, and then we always say to take a core from the side as well. That way you are getting a true representation of the analyte that you are trying to analyze. All right, what's the fourth thing? It allows working with samples safely. It eliminates manual slicing and cutting of tissue. It limits the handling of raw feces. Oh, sorry, clicked it too sure. And it eliminates or minimizes highly toxic aerosols or that come from working with raw feces. Sorry. Oh. Okay. Sample consistency. So our technology and our aliquotters generate and distribute small, uniform, specific volume aliquots in a controlled and efficient manner. So number 6 ensures reliability and reproducibility of the data. Aliquots will thaw faster than a full sample, improving sample consistency and reproducibility of data and results. Samples kept frozen retain the original analyte value because they are not degrading due to the freeze-thaw cycle. Therefore, reaccessing of that same sample will produce the same original results. And lastly, it's a savings in terms of time for scientists. Time savings for improved workflow with a 60-second coin cycle of sample versus thawing out an entire sample, aliquoting, and refreezing. Areas of research that benefit from frozen aliquoting. Currently, most of our customers are working with the microbiome, molecular pathology, biobanking, and bioanalysis. We currently have 3 main products. We have a CXT 353, a 425, and a 450. All of these instruments depend on the type of samples that you are going to be producing and whether you will be putting them into a tube-based, a 96 well plate, or a combination of both. We also do custom instruments. So if the round peg does not fit in the square hole and it does not fit your research, we have designed custom instruments to specifically meet a researcher's needs. So let's go over our analyzers. So the first one is our 353. This is our based instrument. The patent technology provides generated frozen aliquots anywhere from 5 microliters to 300 microliters. It is tube-based only. So the source sample and the destination samples can fit any size tube. What we like about this, it has a laser on the system for pinpointing exactly where you want to core in the sample. The entire deck is chilled by liquid nitrogen. The entire deck gets down to -80. It holds up to 6 to 12 pre-chilled probes. It holds up to 20 different protocols. It has an easy pull down protective guard that protects both the user and the sample. The 425. This one is a little bit bigger as far as a real estate size. The patent technology, again, just like the 353, will generate aliquots anywhere from 5 to 300 microliters. But this one allows for the user to core from tubes, 96 well plates, and will allow for depositing either into tubes or 96 well plates. Again, we can make custom source racks to fit any size tube and any type of 96 well plate that's in an SBS format. It has a laser just like the 353. It holds up to 24 pre-chilled probes. It stores up to 20 different protocols. It has again, an access door with a locking mechanism to protect both the sample and the user. The 450. Again, this is a little bit bigger of a real estate of a machine. This one is designed for using tubes, using 96 well plates, You can mix and match. It also comes with a liquid handling. It has a separate computer for touch screen, which makes it easier for the user to create protocols, to select where in the sample you're going to take cores. Again, it's a 60-second sample core aliquot deposit. It's fueled by liquid nitrogen. Again, the probes are the same. And like I said, this has the capability for liquid handling. If none of those fit your needs, again, we do custom instruments that can be designed and manufactured to meet the customer's needs. This is one of our customers that we designed a custom instrument for them. And that it comes to the conclusion of my talk. We are Basque Engineering and Science, custom system solution and workflows for emerging laboratory research applications. Our distributor is NBS Scientific. We are represented by them here in the US as well as in Europe. If you have any questions, you are more than welcome to reach out to both us and NBS. Thank you. Are there any questions at this time?
Do you have any questions? Thank you so much, Cathy, for the wonderful presentation.
Thank you.
Questions, anyone?
Oh, we have a question.
There's one question, yeah. Thank you
Okay.
for the presentation. If a lot of work upstream to avoid contamination, I have always wondered about a possible cross-contamination between cryotubes stored in liquid nitrogen. I find it hard to believe. According to your experience, what do you think?
So there is no cross-contamination among the tubes. On the 353, one tube is processed at a time. And when you deposit into the carousel, we tell people that you have to deposit and then cap the tube. On the 425 and the 450, it's the way that we do it based on how you set up the tubes. The actual probe, you actually on the 425 have to move, and we tell people there is a way that you move the handle with the probe to eliminate going over other tubes that are on the system. So the way that it will deposit and the motion that we have it set up, it does not go over any other tube. And we've been selling this for over 10 years. I've never had a customer have a problem. And we've done testing where we've taken blot drops where we put white paper on both the samples and on the destinations. And then we do the dye test, and we've never found any cross contaminations among the other tubes. Great question.
Any more questions?
Cathy, could I just ask, what are the, in what areas are people requiring your equipment most often these days? Has it changed? Are there new areas where it's being used?
Yes. So one of the things that has been recent is now with COVID, there are COVID biobanks, and a lot of researchers are working on COVID, and they're accessing samples from 2 years ago when they first started. And then they are doing more studies as they progress. Our instruments fit in a biohood. So the advantage is that they don't have to thaw these COVID samples. So they're preserving, again, the thaw. They don't know if they're thawing the samples, if thawing the samples will play havoc on the virus. So it keeps everything frozen. Plus, with our technology keeping it frozen and no thawing, there are no aerosols. So people that are working with these samples find that that is a safety issue, and that's why they like our instrument. A lot of people, especially with the 425 and the 450, they're using it for their microbiome because working with raw feces is not pleasant. And so therefore, and both that's animal and human. So we have a lot of researchers that are using our instruments to core the feces, because it's easy. The biobanking industry, there's kind of a two-way street here. Most of our biobanking customers are using it more like a QA/QC. So when they get a request from a researcher for a specific type of samples, they want to make sure that those samples are viable before they send them out. So what they do is they take the samples out of the freezer, they put it on our instruments, again, no thawing. They will take an aliquot of maybe 100 megs. They will then run the downstream assay that this specific, researcher is looking for, just to make sure that that sample contains what they're looking for. So that's one way. We do have biobanks right now that are actually working with, they're working with cells, and they're working in a collaboration with some of these researchers, where the researchers will ask the biobanks if they could help them with their assays and help them with, their samples, and they don't want them thawed. So a couple of our biobanks are actually receiving samples from the researcher. They're aliquoting out that researcher's samples and then sending them back in 100, 150, maybe 200 aliquots in tubes and shipping them back to the researchers.
Right.
So there's kind of biobanks are kind of using it two ways. We have pharmaceutical companies that are using it on buffers. One of our biggest customer uses it where they make up different buffers, they freeze them, and then they core into the buffer, and then they deposit them into the 96 well plates. Then they add the reagents, and then they add what they're looking for, and they do a concentration gradient. So biobanks, pharmaceutical companies, microbiome. I'm trying to think of where else our application. Animal is huge right now. So just for an example, we have 2 customers that are doing studies on golden retrievers and labs, because they tend to get cancer at a young age, unfortunately. And so they're using our instruments and our technology because they have multiple frozen dog feces that they need cored. And thawing that many samples, it's just not feasible. So our instrument is quick and easy. It's safe, and for most of our customers, they love it. It's so easy to use. I'm trying to think of other people that that's probably the majority. We have a few people in the tissue industry that use it for cancer research. So when they do a biopsy and they flash freeze it, they bring it down and they can pinpoint immediately the dark spots within the tissue. and they can take that core and immediately analyze it. We do have an instrument outside of a cardiac wing that's exactly what they do, that as soon as they're doing biopsies, they flash freeze it, and then they bring it down to the lab and they do their analysis. So a lot of tissue work, a lot of cancer. So that's really the four main. We do have some people using it for metabolites, because obviously some of the metabolites are very labile. So anybody that's doing metagenomics will use it for their studies, and they basically are using serum and plasma. So and cells is brand new. So we are working currently on a very viable solution for cells, because people want to try to their cells is like gold, so they want to retain as much of the main sample as they can. So we're working with a lot of people on that as well.
Okay. Thank you so much for the answer. I mean, it's really interesting and a wonderful presentation.
Thank you. Thank you.
So